In December of 1993, Sir Richard J. Roberts was awarded the Nobel Prize in Physiology and Medicine for the discovery of mRNA splicing, a fundamental discovery that redefined our basic understanding of how human and other eukaryotic cells work. He specifically found that genes in eukaryotes are not contiguous strings, but rather contain introns, or nucleotide sequences within a gene, and that the splicing of messenger RNA to delete those introns can occur in different ways. This, in turn, yields different proteins from the same DNA sequence. This discovery, and Sir Richard's subsequent work, have had a far-reaching impact on the global biotechnology industry and many other fields. For example, RNA splicing is crucial for understanding the human genome because it significantly expands the protein diversity produced from a limited number of genes. In this and in many other ways, his work continues to shape science and the world around us. Sir Richard began his journey to the very pinnacle of scientific achievement in the UK. He was born in 1943, the only child of John and Edna Roberts in Derby, England. One of the earliest hints of his tremendous scientific future came when his headmaster in elementary school saw the early potential of Sir Richard. He encouraged him to solve challenging mathematical and logical problems. This inculcated in him a lifelong interest in mathematics. His next major formative experience came when he was given a chemistry set. His father supported his interests and arranged for the construction of a makeshift lab. Sir Richard was so fascinated at that early age that he thought he must become a chemist. After passing the A-level exams, he was admitted to Sheffield University, which was his first choice on account of their excellent chemistry department. After his undergraduate, Sir Richard became a PhD student in the lab of Professor Olis, and he began to explore various compounds found in the heartwood of various tropical Brazilian trees. He soon came across a book by John Kendrew that was to change the course of his latter research career. It described the early history of crystallography and molecular biology, focusing on the prestigious MRC laboratory in Cambridge, UK. It was his first exposure to molecular biology, and he knew immediately he wanted to pursue this. For postdoctoral studies, he was offered a position at Harvard by Professor Jack Strominger. And so it came to pass that on a frigid January day in 1969, he and his family arrived at Logan Airport to start a new life in the US. He was soon assigned with the job of sequencing a transfer RNA that was involved in bacterial cell wall biosynthesis. In 1969, only a handful of such transfer RNAs had ever been successfully sequenced. After careful study, Sir Richard decided that a new method, pioneered in Fred Sanger's lab in Cambridge, UK, was a better method. So in late 1970, he set off back to the UK to learn these techniques. On his return to Harvard, his small sequencing operation was the first in the Boston area, and many researchers came to learn these techniques. When his postdoc time at Harvard came to an end, he was offered a job by none other than the Nobel Prize winner James Watson, the co-discoverer of the structure of DNA. This occurred after a 10-minute meeting, during which he mainly listened. In September 1972, he moved to the world-famous Cold Spring Harbor Laboratory on Long Island. He soon became involved in the production of so-called restriction enzymes that could cleave DNA into specific pieces. This was to shape much of his subsequent research career. He began a systematic search for new restriction enzymes. His growing enzyme collection was a valuable resource to scientists around the world. He constantly sent samples to other researchers and was inundated with visitors. Fully 3/4 of the world's first restriction enzymes were discovered or characterized by Sir Richard and his colleagues. This contribution to the development of global biotechnology cannot be understated. In a move that would eventually lead to his Nobel Prize, he began to map the DNA of the adenovirus. All seemed to be going well until he and his group came up with the startling finding that all late mRNAs seemed to begin with the same capped oligonucleotide, which was not encoded on the DNA next to the main body of the mRNA. He had excellent biochemical evidence for this, but the real proof was elusive. Then the critical breakthrough came in 1977. Collaborating with talented electron microscopists, he and his team were able to directly visualize the mechanism of mRNA splicing. Sir Richard immediately realized the significance of this discovery, and he knew it was worthy of a Nobel Prize. After these heady and impactful discoveries, in 1992 he left Cold Springs and moved to New England Biolabs, a small private company of 150 researchers making reagents, restriction enzymes, and carrying out basic research. He is presently the Chief Scientific Officer, and he continues his work to this day. Sir Richard Roberts continues to do critical work that still shapes and influences the future of biotechnology, as well as our fundamental understanding of basic processes in molecular biology. A true pioneer, he will continue to inspire many generations of young scientists as they seek to extend and build upon his monumental contributions.
You know, I became a scientist in I think a little strange way in some instances. So when I was very young, I loved mathematics. And I went to a primary school and then a junior school. And at the junior school, the headmaster used to give me puzzles to do every so often at night as I was walking home. And I thought he gave this to everybody, but it turned out it was just for me. But these were all mathematical puzzles, and I just fell in love with mathematics. And if you'd asked me until probably age, 9, 10, I would have told you I was going to be a mathematician. This was what my life was headed for. And then I read a book called How I Became a Detective, a regular sort of, criminal kind of detective. And after I read that book, I decided I was going to be a detective. And then my father bought me a chemistry set, and I discovered chemistry, and it was in the days when the chemistry sets were interesting. You could actually do things with them that were things that you couldn't do today because they've tamed these chemistry sets, so they're boring these days. But I discovered I could make fireworks, I could make explosives, and I fell in love with chemistry. And that lasted with me. I did chemistry at school, I did chemistry at university, at Sheffield University, and until the very last year of my PhD, I would have told you I was going to be a chemist, and this was going to be my life. Then I read another book by a man called John Kendrew, who won the Nobel Prize for protein structure, but the book was about the origins of molecular biology. It was called The Thread of Life. And I would say by the time I finished that book, I knew I was going to be a molecular biologist, and that's what I ended up being. But the nice thing is what I do these days is a mixture of mathematics, chemistry, biology, molecular biology, and computing, because I figured out how to write computer programs many, many years ago, and I really do bioinformatics all the time. I look at the computer screen and make discoveries that way these days. So my life as a scientist has changed quite a bit over the years, but now finally I'm sort of doing everything that I loved doing when I was a child, so it was great. Yes, it's very much a synthesis of my entire life. I think one of the things you find when you're doing science is that you make discoveries all the time. It's not just that there is sort of 1, 2, 3 discoveries that stand out and you say, oh, this was great. You make discoveries all the time. And so when I first started doing my PhD in organic chemistry, I discovered a compound that my professor had predicted would be an intermediate in a biosynthetic pathway. And that was fantastic. This was really good. He was really happy. He had a number of other students who'd all been looking in different what I was doing, I was studying the heartwood of a Brazilian tree. And There were others who'd been looking at different trees, and they hadn't found this intermediate, and in my case, it was stable, which is a good example of the luck that I've had throughout my life. I tell you, luck has played a huge part in my life, but it plays a huge part in everybody's life. And the secret is to take advantage of it, of course. But because of this, by the time I had done 1 year of my PhD, I could have written my thesis. I had all the results I needed. Then I went, eventually after my PhD, I went to Harvard, to Jack Strominger's lab, and there started working on transfer RNAs. These are small molecules that transfer amino acids to the ribosome when you're making proteins. And it turned out in the organism I was looking at, one of these tRNAs was not involved in transporting amino acids into proteins. It was taking the amino acids and putting them into the cell wall, the bacterial cell wall. And there was one tRNA that had been specialized for this purpose. It didn't work in protein synthesis. It only worked in cell wall synthesis. And I ended up getting a Nature paper out of that. So, that was a really nice discovery. But the thing that really happened was that I learned then how to sequence RNA using methods that had been developed by Fred Sanger at the MRC lab in Cambridge. I went over to his lab, spent 6 weeks, learned how to do it, came back to Harvard, and I was the first person in the Boston area to be using Fred Sanger's techniques to sequence RNA. And so loads of people came to me to learn how to do it. I met a lot of people in those days, many of which are still good friends of mine. But that was another piece of luck. I would never have predicted that this was going to happen. That led to my getting a job at Cold Spring Harbor because Jim Watson, who was one of the discoverers of the structure of DNA, I was told he was looking for someone, that I was the perfect person for him. I went, I had about a 5-minute, 10-minute interview in his office. He said, I got a job for you at Cold Spring Harbor. And it wasn't an interview. He just said, I got a job for you. And I thought, well, that's a little odd. But anyway, so I ended up going to Cold Spring Harbor. But when I got there, I discovered that the particular method using RNA to copy DNA in order to determine the sequence, that this approach was already being used to sequence SV40 DNA, which was a small oncogenic virus that people were interested in. There were 2 groups already doing it. So why would I do it too? So I started doing something totally different. Of course, that led to Jim Watson wanting to fire me for not doing what I was told. But that's another story. But what I did, I'd been to a lecture at Harvard just before I left by Dan Nathans, who was describing what you could do with enzymes called restriction enzymes. These are enzymes that cut DNA at very specific locations and make small pieces. And so I decided that, well, maybe one of the reasons that RNA sequence had been developed ahead of DNA sequencing, because there were small RNA molecules you could practice on, but no small DNA molecules. And these restriction enzymes offered the chance to make small DNA molecules. And so I thought, well, I'll develop DNA sequencing methods on DNA directly, not through RNA. But then we started looking for these restriction enzymes, and we just found more and more all the time. Very soon, we discovered 30 of these enzymes, and it turned out everybody was interested in them, not because they wanted to use them to sequence DNA, but they wanted to use them for cloning and so on. And they became the start of the biotechnology industry. Everybody wanted these enzymes. We were giving them away. We were sending the strains out, telling people how to make them. And they became of great interest in themselves. But Jim Watson, again, thought that I was wasting my time doing this. And so we had some constant themes about that. But when postdocs used to come to my lab, I would say, well, first find a restriction enzyme, because we know we'll get a publication out of that, and then do something that's a little more speculative, something that isn't going to be, quite so mundane, but that you don't know whether it's going to work or not. And so one of those projects I gave to Richard Gelinas, and his project was to ask whether promoters, sequences in DNA that tell an RNA polymerase where to start making RNA, was that sequence the same in higher organisms as it was in bacteria? And it was in the process of doing that that we discovered RNA splicing. That was a huge discovery, was really a wonderful discovery to be made, but it was made because the experiment that I devised that was going to help us tell where the promoter was didn't work. It just didn't work. Now, what I discovered then, and something I always tell students now, is that failure is a good thing, because if you find out why you fail, either it's because you screwed up, maybe you dropped the tube on the floor and picked it up, or you just did something wrong, or maybe it's because nature is trying to tell us something. And in this particular case, nature was trying to tell us something. It was telling us that the way in which higher organisms make RNA is different from the way the bacteria make RNA. The basic process is the same, but in going from the initial transcript into the ribosome, eukaryotes cut and splice that RNA in much the way that a film editor copies and edits and cuts and splices movies when they're trying to make them. You know, they take a series of scenes, a lot of separate cuts, and then they cut and splice to make something that looks as though it's just a continuous action. And that's how eukaryotes make the RNA that goes to the ribosomes and make all the proteins that keep us alive. Yeah, well, I think one of the things about mentors is that they can play a very important part of your life. And sometimes it can be good, but sometimes it's not so good. So, you know, one of my first mentors that I had was the headmaster at my junior school, who gave me these puzzles. He saw something in me that I didn't know was there. And so he turned out to be very important, and it was fortunate he died just after he heard that I'd won the Nobel Prize, and so I was rather happy about that. He was a very nice man. The next mentor I had that was really good was a postdoc who was in the lab in Sheffield when I was looking at this heartwood of a Brazilian tree. His name is Kazu Kurosawa. He's a Japanese gentleman. And he was one of these people who could tell you what to do, why you were doing it, and then help you to understand the results. And he was just really good at this. And so I learned more chemistry from him than any textbook, anything I ever did. He was just great. He also taught me how to play Go, a wonderful Japanese game, and several other things too. And after he left and went back to Japan, we used to play Go by mail. Can you believe that? This was before email. We used to play Go by mail. We would send moves to and fro for several years until his English got so bad he couldn't write the letters that came along with it. So that was good. And then I went to Harvard, and there I didn't really have a mentor in the traditional sense. Jack Strominger, who I worked for, had been a professor in Wisconsin, worked on cell wall biosynthesis, and had just been newly appointed as a professor at Harvard, which was his lifelong dream. And he chose that moment to change fields and do something different. So I picked up a project from a postdoc who was there, and he was trying to sequence this tRNA, but he was doing it by one old-fashioned method. And I was reading about all of this and found a new method that Fred Sanger had developed and said, well, I'm going to go off and do that. And so the postdoc, Tom Stewart, was only there for a short time. He left, went back to Australia. I was left on my own. And Jack really let me do what I wanted to do. And so that was very nice. And then went to Cambridge. Fred Sanger learned how to sequence RNA and came back to Harvard. And then went to Cold Spring Harbor. And the person who should have been a mentor there, Jim Watson, was really not at all, because we used to fight all the time. So I spent 20 years fighting with Jim Watson at Cold Spring Harbor. But I was lucky because I started working on these restriction enzymes, and a professor at Rockefeller called Norton Zinder was very keen on these restriction enzymes and thought what I was doing was very good. And he was Jim Watson's friend. And so every time Jim Watson threatened to fire me for not doing what he wanted me to do, I would talk to Norton, and he said, "Oh, don't worry, I'll talk to Jim." He would talk to Jim, and then Jim wouldn't fire me. So I lasted for 20 years that way. I think a lot of people want to know what it felt like when you make this discovery that may lead to a Nobel Prize. Well, for a lot of people, they make that discovery, but they don't talk immediately about Nobel Prizes or anything like that, because very often the discovery itself is not immediately Nobel worthy, shall we say. You have to wait until there's been some further developments, and all of a sudden you realize that this could be Nobel worthy. But in my case, we made the discovery we produced the experiment that actually led to the data that proved what was going on in the morning. And by the afternoon, everybody was talking about Nobel Prizes because it was such a big thing. And it was one of these things where previously we'd been saying that we thought this was the way that RNA was being made in higher organisms. No one really believed it. They said we were looking at an artifact, so why waste our time doing this, and so on. But then this experiment proved that what we'd been doing for a year really was true. And so at that time, it seemed pretty good. But then almost immediately when we started talking about it, other people tried to claim that they had made the discovery. And so there was a huge amount of I don't know quite how to put it, but there were a lot of people who wanted to have the world believe that they made the discovery because they all knew that there was a Nobel Prize potentially here. And so that was kind of disappointing to me in many ways because, I knew who'd made the discovery, but eventually it led to the right outcome, so it was all okay. But it can be a little disconcerting. But, the worst is to know that you've been nominated for a Nobel Prize and then not to get it. And so in 1977, when we made the discovery, it wasn't until 1989 that I knew I'd been nominated. And I was pretty sure that, I was going to win it. I was absolutely sure. And then when I didn't win it that year, the disappointment was unbelievable. And so it's really better not to know. It's absolutely better not to know. We can talk a little bit about GMOs and food security and how to improve the crops that are around. We've been breeding crops for some 10,000 years, crossing them, trying to improve them, trying to bring better traits into them. But then there was a major breakthrough that came about 35 years ago when Mark Van Montagu and Jeff Shell discovered how to transfer genes from a bacterium into a plant. And this is something that takes place naturally. But they discovered how the natural process worked and then realized that you could use this not to put the gene that the bacterium wanted to put in, but rather to put in the gene that you wanted to put in. So, if you wanted the plant to grow taller, you wanted it to produce more grain, you wanted it to have better nutritional value, you could put in appropriate genes using this mechanism that Van Montagu and Shell had discovered. That was a huge insight, and it really made the possibility of improving plant breeding in ways that were unimaginable before. The method that they chose would put genes in, but you didn't know where they went. And so in some cases, they would not lead to improvement in the plants. And so you had a lot of work to do to figure out what was the best plant. And what happened was that a group at Monsanto started using these techniques to improve plants. And Greenpeace decided that this was an opportunity for them to stop Monsanto putting food onto the European market, stop bringing seeds and things into Europe. And they used this as an excuse to start a campaign against GMOs. And it's unfortunate because there was at the time not enough science to know that they were safe. And so it was reasonable to ask that, one should do some science and so on. But the consequences of that have been that they're still on this same boat because it's been very profitable for them. As soon as they took this anti-GMO campaign, they tripled their income in Europe. And they've just kept at it. And even though we now have 35 years of experience, not a single problem, not one, associated with GMOs. We now have better methods of doing it thanks to CRISPR and this technology, which allows you to be very precise in where the gene will go and how it's going to go in and so on. And the problem is that whereas we in the West don't need a lot of genetically improved plants, we've spent so much money on agriculture because it's a very profitable business in the West. But in the developing countries, where they've not had the same improvements possible in their plants, because the agricultural companies didn't want to do it, they can't make money that way. And so they could use these CRISPR methods, these new methods of gene editing, to greatly improve the plants, but Greenpeace have gone out and told them that they shouldn't do it and that they're dangerous. And they're basically selling misinformation about this. I would say disinformation. They're deliberately saying that these things are dangerous. And I find this personally to be almost a crime against humanity. I find it awful. And so I've been mobilizing the Nobel laureates. We've been speaking out in favor of GMO plants. We have 179 Nobel laureates signed on to a petition to say that GMO plants are okay. And we're trying to persuade governments that they should approve these, that they should accept them and not listen to Greenpeace. I think one of the things that I've always carried with me in my life is that luck has been incredibly important. And I learned a very important lesson when I think I was probably 17. I was very good at snooker. Billiards and snooker is sort of what they play in the rest of the world instead of pool. But I was really good. I was the West of England junior champion at snooker. And I was playing in a tournament one day, and I'd made a shot, and I'd hit one ball, tried to pot it, but instead my ball bounced off and potted another ball. And when I came to play the next shot, I muffed it, and it just didn't work. And afterwards, an old gentleman came up to me and he said, you know, you could be a really good player, but you have to learn that when you have a piece of luck, you have to take advantage of it. If you don't take advantage of it, what was the point in having the luck in the first place? And that has really stuck with me throughout my life, that I really every time something lucky happens, I try to find a way that I can take full advantage of it to move on to the next. You know, this whole business about how important luck is you have to recognize everybody has it. Okay? And if you don't take advantage of it, well, what was the point in having it? And too many people have a piece of luck and then they feel guilty about it. They feel, oh, well, you know, my friend over here didn't have luck. Why should I take advantage of my luck? And you really should take advantage of luck. I think it's incredibly important. And then the other thing is the thing I'd mentioned before about failure, that failure is actually a time when you learn. People who never fail don't learn very much at all. And I was talking to some parents one time because parents often tell their kids, well, you know, if you fail, you're no good. And teachers will say, oh, if you fail, you're no good. And they view failure as being a problem. It's not. Failure is when you really learn, provided you go back and figure out why you failed. And this is why flying is one of the safest ways of traveling around the world, is because every time an aircraft comes down, there's a massive post-mortem done to find out what went wrong, and then they can fix all the planes after that. Doctors don't quite do the same thing, you might be aware. They don't like to go back and find out where they failed, and they typically do not want to tell you why they failed, because they feel that for them, a doctor-patient relationship is about trust, and if you tell them, oh, I shouldn't have given you that medicine, then the patient will lose the trust in the doctor instead of admiring the fact that the doctor was honest. And said exactly what happened. And it's the same when you're a scientist and you're doing experiments. If they don't work, really be very careful looking to see what went wrong. I love it when students come to me and said, oh, you know, this experiment you told me to do, I did it and it didn't work. And I always think, great, you know, maybe nature's trying to tell us something here. Maybe there's a discovery to be made. So failure is a good thing. It's when you really learn. Again, a piece of luck, a discovery. So when I did finally win the Nobel Prize, one of the things you have to do is you have to go to Stockholm, pick up the prize, meet the king and so on, but you have to give a lecture, the Nobel lecture, and you get the committee there and all the scientists and so on. And most people will talk about all the work that they've done and the discoveries that they've made following their initial discovery. But I'd actually left the field many, many years previously, and I'd started working on DNA methylation in bacteria. And one of the things we'd been trying to do for quite a long time was to get a crystal structure to know exactly what happens when a DNA methyltransferase binds to DNA. And just 2 months before I had to go to Stockholm to give my Nobel lecture, we made a big discovery. We'd discovered that when this methylase binds to DNA, the base that it's going to methylate is flipped 180 degrees out of the helix into the active site in the enzyme, in the DNA methylase, gets methylated, and then flips back in. And it turns out this property of flipping bases out of DNA is something that quite a lot of proteins use, not just DNA methylases, but this was the first time that it had been discovered. So it always seemed to me this was a good topic to talk to the Nobel Committee about ahead of time. So that was the basis for my talk when I gave my Nobel Prize speech.